Well, okay. Good morning.
Good morning.
Good morning.
Yeah. Well, we are ready for a treat, right? We have Dr. Liang that he's going to teach us something about fluorescent spectroscopy and protein purification. And so I hope you will enjoy it and take advantage of learn everything that you can. Under these circumstances, the best way will be hands-on, but this is what we have now. So Dr. Liang?
Hey, Dr. Renkang, I cannot share my screen. It's a host disabled participant screen sharing.
Aha, okay.
Sorry about that.
Well, I didn't know that part, okay.
Yes.
So, I guess, where do I do that?
Maybe it's in the setup for your account.
Uh-huh. Okay, I think, I wonder if...
Maybe try to figure out where can I find it too.
Okay, I wonder if I select you here. Because, I remember doing this in class. The students
Right.
did a presentation.
Right.
And okay, I found it.
You found it? Great.
Yes.
Thank you.
So I'm going to make you, but I don't think I did it that way. But we'll do it that way. I'm going to make you the host.
Yes, please.
Okay, here it is. And now I know where to find that. Okay, change host. And now you are the host.
All right. Thank you so much.
And it's recording.
Excellent.
Yeah.
All right. Good morning, everyone. Sorry for the delay a little bit. So can everyone see my screen right now?
Yes.
Yes?
Yeah.
Yes, good. Sorry about that. It's earlier morning, and I know everyone already familiar their schedule, and then this is going to be a hard time to wake up earlier, right? But I like to start. So today I'm going to talking about the principle of the fluorescence, spectroscopy, and the protein purification. So usually what I do is I will give it some principle in the morning, and then roughly in the afternoon I do some experiment with students. But unfortunately, we cannot do anything right now, so I just added the other extra topic, which is protein purification there, and we'll see later on that. So my name is George Liang. I'm from the chemistry, and then I'm a biochemist, right? So basically, I'm using a lot of biochemical and biophysical methods to study the protein-protein interactions. All right, I hear some noise there. I don't know what is that. So anyway, the part one, I'm going to talk about some theory of the fluorescence first. All right. So go back to 1672, long time ago. We'll talk about the light. All right. So in Sir Isaac Newton in 1672, he showed that the component of color of a visible portion of the light can be separated through the prism. As you can see, those light go through the prism, they were able to separate out to different color on that. And then these kind of light were able to bend, which they call the refraction later. And then in a different degree according to the wavelength. And then he also developed the theory of light, which is they thought is a corpuscular of the light. Basically, it's a particle of the light. So light is a particle in that time has been proposed by the Newton. But in the 1692, the Christian Huygens also proposed that the light basically is a wave theory of light. So light in that time, they believe that is a wave, all right, it's not a particle. So you may already took the gym camp, there is a two argument on that. Some people say light is a particle, some people say light is a wave. And then the question is, what is that? So until like 1905, Einstein, he won a Nobel Prize for the physics, and then he proposed a photoelectric effect. So basically, his work proposed that the light can be considered as consisting of particle called the photons, and also he can consider as a wave as well. So he also stated that the energy the photon contain is proportional to the frequency of radiation. So basically, light is not just a particle or it's not just a wave, it's a combination of both. It can consider as a particle, it can consider as a wave altogether. And that's why you can see there is a mechanical wave for this light, and also there's a light wave at the bottom, and then basically this combination of the particle property and the wave property. And of course, I'm the biochemist. I like to understand how the molecule going to interact with this photon and how the photon going to change with interaction of the molecules. And of course, go back to the topic here, what is fluorescence? Why we know there is a fluorescence for some molecule, why some molecules do not have fluorescence. So basically, fluorescence is one of the intrinsic property for some molecule. And this molecule, as we know, that most of molecule based on the Boltzmann distribution, they are always stay in the ground state, which is on room temperature. Most of molecule is in the ground state. But in the presence of light, and this light could be some energy source, and this light will able to absorb by the molecule, which you can see the green color in the ground state. And this molecule, once they absorb the light, they will move up into the excited state. And this excited state we highlight in those kind of orange color 9. And there's S1 prong or S1. So S means a singular state, but there's S1 meaning there's excited state. And this excited state, because molecule they undergoing absorb the light, those electrons in the molecule will move it up, they will vibration or moving around, and then they will able to change this electron configuration. That's why they go to the excited state. But interesting that this molecule is not going to stay in the excited state for a long period of time. Over the time, this molecule will go through the first vibration level of the excited state. We call this S1 state. And this S1 state, they are not going to just disappear. This excited state is going to release energy because they absorb energy from the light. So therefore, they are going to release energy out of the molecule. And this energy release, they call this is a fluorescence. So fluorescence basically is a molecule that absorb the light, and then they go to the excited state, and then from excited state, go back to the ground state, and this can release the light again, and this we call the fluorescence. And why we want to study fluorescence, right? You may wondering why, because it's pretty, as you know, they're different color. You can see the test tube, you can different color on that, and that's why it's very easy to observe by the chemist. So basically, it's pretty, and also it provides information on the molecular environment. So we will see later on fluorescence is highly environment sensitive. They are very sensitive to maybe the ion concentration, or they are very sensitive to your electric field. And also they are very sensitive to viscosity, polarity, pH, and temperature. So basically, scientists, they can use in the fluorescence as a way to monitor the environment change. And also it provides information on the dynamic process on the nanosecond time scale. So fluorescence, basically, as we already see the previous slide, they found ground state to excited state. And then from excited state to ground state, this kind of transition is very, very fast. All right, it usually happens in the nanosecond or microsecond range, which is very, very fast. And also fluorescence is very, very, very sensitive. Right, so it can work with the sub-nanomolar concentration, and then basically it's kind of like femtomole concentration. All right, femtomole basically is a 10 minus 15. All right, so it's a very, very small quantity, and then you can detect the molecule under very, very, very low concentration, and that's why I say it's very, very sensitive. And also it's even like single molecule study. They can able to using the fluorescence as a way to study in a single molecule. So anyway, the fluorescence nowadays has been very popular, all right? They involve a lot of technique, and we're going to give you some of the examples for those techniques later on. All right, so since I cannot demonstrate anything here, I would like to show you guys a video, and then you guys can see how this video looks like. So basically, I usually would do is I will bring anti-freezer. So in your car, right, you will load in some anti-freezer, and those anti-freezer basically they add some fluorescent dye inside. Usually in the class, I will bring anti-freezer solution, and I shine the UV light on it, and you can see some color show up. And in this video, they also want to do that as well, but they are going to show you guys the tonic water. And so tonic water is the one you can see in the club. And then when you shine UV light on it, those tonic water will show the fluorescence because they have some chemical inside. So let's watch this video right now. Hopefully it's going to work. Okay, here we go.
Dr. Liang, I don't think
Yes.
we can see it because yesterday Tina was trying to show us a video too, and it wasn't sharing on our screen. And if you played it, we can't see it right now either.
Oh, you cannot see that either?
No, we're just seeing the bottle. I mean, that at least on my screen, that's all I see. I can't see everyone.
Let me see if I can find the other way. So maybe I will go to this website to see if can you guys see that right now for my website?
We still see the PowerPoint right now.
You still see PowerPoint?
Yeah.
Okay. Oh, I think hold on. Let me see if I can do the other again. Share screen. So
maybe if you get all the way to the video and then share the screen from that website, it might work.
Yeah, I think I know how to do it. I need to share the Chrome. Can you see that right now?
Yeah,
can you guys see that?
That's working. Yeah, that's working.
Okay, good. It's very short, only like I think only like 5 minutes or 3 minutes video. All right, so it's a very short video. All right, so you guys ready? Let's give it a shot. All right. So now we know that fluorescence is everywhere in our daily life, right? So let's go back to the slide. Can you guys see the slide right now?
Yep, all good.
Okay, good. So basically, as you can see on that fluorescence, basically some molecule, they absorb the light and then they produce those, different color or visible color light, and then we were able to see by our eyes. And then of course, as you can see the video, there a lot of application has been used nowadays. So they're also talking about the fluorescence. In the beginning, you need to have a molecule to absorb the light first. And in order for the molecule to absorb the light, we usually use in a machine called spectrophotometer. And this spectrophotometer is a machine, I know you guys in the Gen Chem or maybe in the organic chemistry, you may already do this. So basically, you will have a sample, like in the cuvee, you can see those green color cuvee. You put some of your sample into this cuvee, and then you put it into the machine, and machine has a light source, and this light source can go to the prism, and this prism can, of course, can split out different kind of wavelength of the light. And then this light will go to your sample, and then they will have detector after your sample. And they measure the decrease of your signal as a way of saying that how much light has been absorbed by your sample. And this is basically the spectrophotometer. We already know on that. And then, of course, this is based on the Beer's law, and you already hear this from the Gen Chem. Maybe A is absorbance, epsilon is excitation coefficient, B is a light path, C is a concentration. So this meaning that if you have a higher concentration of your sample, you are going to have a higher absorbance. All right, so it's a linear proportion between the concentration and absorbance. And this basically just measure the absorbance. All right, this is not measure the fluorescence here. They just tell you that how much light has been decreased in the presence of this molecule into your cuvee. And of course, the other way to detect the fluorescence, we call this a spectrophotometer. All right, so absorbance we call spectrophotometer. Fluorescence we call a spectrophotometer. Sometimes people call it photometer as well. And then, of course, the way they set up is a little different than the absorbance setup. You can see there is a light source, which is the same as before, and then it can go to the wavelength selection. And you put a sample into the sample holder. But in this case, instead of monitor right after your sample, they monitor about 90-degree orientation of your detector. What does this mean? Meaning that your fluorescence is coming from nothing to something, all right? Because you were not able to see the light in the beginning because basically it's a 90-degree measurement. As compared to absorbance, as we see the previous slide, your absorbance is measuring the decrease of your light intensity, all right? That's why there's absorbance. But fluorescence, basically the light source will not go to your sample, they just go straight in, and then you detect the 90-degree. Therefore, your signal will be very, very, very sensitive because it's coming from nothing. Because originally, at a 90-degree angle, they should not have any light source there. So that's why I keep saying that fluorescence is very, very sensitive, the signal from the molecules. And also, there are two different types of fluorescence measurement. One is called emission measurement. The other one is called excitation measurement. So emission basically is the molecule transition from the ground state to the excited state, and once they are in the excited state, they will release the light, and this is called emission spectrum. So basically, emission spectrum, they will measure the same as we see before. There's a light source there, and then there's excitation wavelength selection, and then they shine the light into your sample, and of course, your sample going to produce fluorescence. And then there's 90-degree detection there, and then in the detector, you will see those peak here, and this peak represent your fluorescence as a function of your wavelength. And basically, this is emission spectrum, so they were able to measure the fluorescent emission for a fixed excitation wavelength. So you have different wavelengths shine the light in, and then they will show different kind of the fluorescent signal as output on that. And the other way is called a fluorescent excitation spectrum. So excitation, emission, basically they are different, all right? But excitation is the same as absorbance, but it's just not 90-degree. It's not a 180-degree measurement, it's a 90-degree measurement, all right? So excitation, basically what they're gonna do is they will measure the fluorescence emission spectrum at the fixed wavelength while scanning the excitation wavelength. So they are going to scan in this excitation wavelength. That's why you can see this wavelength here in the x-axis, and the y-axis will be your fluorescence signal, all right? So you are going to tell the detector focus on a specific emission wavelength, and then the system will scan all the excitation wavelength, and that's why they show up this kind of peak. And basically, people believe that excitation is very, very similar to the absorbance spectrum. But definitely, emission will be opposite, right? Emission will be monitor different kind of excitation wavelength. Like you can see number one here, they change the excitation wavelength, and then they were able to look at what is the emission spectrum on that. So, as I said in the beginning, we cannot do any experiments. So just let you guys know, basically, most of the fluorescence was measured by the fluorescence emission spectrum, all right? And of course, emission spectrum and excitation spectrum, usually they will form like a mirror image. As you can see here on the black solid 9, this is emission spectrum. As compared to the dash 9, this is excitation spectrum. And of course, in the y-axis is fluorescent signal, in the x-axis is wavelength. All right. So the reason why they will form the mirror image, we will see in the next few slides, and hopefully can expand on that. But basically, people always report the fluorescence emission spectrum as a way to represent this fluorescent signal from this particular molecule. All right. So, since we're talking about this fluorescence emission spectrum, and of course they have like 3 general rules about this fluorescence emission spectrum. All right. So in the rule number 1, they say in the pure substance is in a solution in a unique form, the fluorescence spectrum is invariant, remaining the same independent of the excitation wavelength. So no matter how change different kind of excitation wavelength, their emission spectrum is always the same, all right? And the reason for that, because we will see in the next slide, right? There are some energy diagrams show up, and they was understanding why we change different excitation, how come they still get the same fluorescence signal on that. And the second general rule is that the fluorescence spectrum lies at a longer wavelength than absorbance spectrum. And sometimes they call this a stock shift. So we do see here this the mirror image between emission and excitation, and the one we're talking about fluorescent emission spectrum, as you can see under the maximum, the highest intensity of this emission spectrum is always has a higher wavelength than the excitation spectrum, and that's the one we're talking about. The difference between this the peak here, basically there is called a stock shift, all right? And this stock shift is an example of this why the fluorescence always had a longer wavelength than absorbance. And of course, I talk about this, you guys have still no idea why it's on that, because we'll see those in the next few slides, right? And the third general rule is that the fluorescence spectrum is, to a good approximation, a mirror image of absorbance band of this frequency. And we already saw on that. So basically, the emission spectrum is a solid 9 here, and absorbance spectrum you can consider same as a dash 9 here is excitation, and they form a mirror image with each other. All right, and the reason why they will form a mirror image, again, I'm not going to tell you guys right now. We will see some of the information in the next few slides. All right, so we are going to look at one at a time of these 3 general rules, and then I'm going to explain again about why this general rule is going to apply into this fluorescence emission spectrum. So the first one is why we change different excitation, they always get the same emission. All right, so go back to this kind of energy diagram in the right. So before I talk about this, I just want to give a little bit explanation first. So S0 meaning that is a ground state, all right? Those black 9 is a ground state. And of course, you can see the solid black 9 is very, very stable ground state. And then the dash black 9, meaning that those are kind of different vibration level of the ground state. And of course, S1 state is excited state, and S2 is a further excited state. So they have different kind of excited state for this electron. All right, so if there's a molecule like fluorophore basically is a fluorescent dye, all right? And this fluorescent dye, they were able to, in the presence of light, they were able to move from the ground state to the excited state. And usually they will, for example, you shine the light into the a pink color one, and then they will transition from the ground state all the way to the S2 state, all right? So it depends on, which molecule you're talking about, also depends on the light intensity you're talking about, all right? So we cannot predict the molecule going to which excited state, we don't know, all right? So once they go to the shine the light on the molecule, once they go to the excited state, and this molecule, is undergoing those kind of thermal relaxation, or called internal conversion. The internal conversion basically is not going to emit any photon, all right? So those basically just release as a heat. So when they're in the excited state, they maybe release this kind of energy as a heat, and they can slowly, very, very quickly, like 10 to minus 12 seconds, migrate into the first excited state, first vibration level of the excited state here, which is the solely red color S1 state. And once they hit this kind of the lowest vibrational level of the first excited state, then those molecules has no way to go, all right? And the only way they will go is they will release as a fluorescence, all right? And that's the reason why you excited, so for example, you exciting the pink color one, they go to the S2, you excited those blue color one, they go to S1, you excited light blue, they go to the other S1. So you have 3 different kind of excitation wavelength, they go to 3 different kind of excited state. But no matter how they excited, the bottom line is those molecules going to internal conversion all the way to the lowest vibrational state, the lowest vibrational level of the first excited state, which is S1 red color 39 here. And when they hit this point, they will release as fluorescence. And that's why I'm saying that no matter how you excited on it, they always show you the fluorescence, the same wavelength, all right? And that's why they say this is a fact account for the independent of the emission spectrum from the excitation wavelength. All right, so does everyone got it? I know this may be challenging, all right? Some of you guys may see this in the PChem or the physical chemistry. Some of you may not be able to see that forever, all right? So the bottom line is no matter how you excite the molecule always show you the fluorescence emission in the same wavelength. And of course, if you excited at the pink color, definitely you may have a need to require like high energy on that, as compared to shine the light into like light blue color, your excitation energy may be a bit lower than that. All right. All right. So this is the first principle, or first the phenomena property for the fluorescence. And the second, sorry, still the first. All right. So they still want to show on that, if you excited, so on the left here, this is excitation spectrum. On the right here, this is emission wavelength. And of course, you excited, for example, excited this kind of maximum excitation, you got see this kind of emission show up. But if you excited this kind of like shoulder of this mountain, you can still see they have the same kind of emission there, although it's just a little bit lower intensity than you excited at a higher excitation energy there. So the only difference is intensity will change, but the bottom line is no matter how wavelength you excite the molecule, the emissions maximum wavelength will be the same. All right, so this is kind of the spatial feature for this kind of fluorescence emission spectrum. And the other the phenomena here we're talking about is the fluorescence usually lies at a longer wavelength than absorbance. Right, we know they can form a mirror image within each other, and then one of them usually has a longer wavelength. And the question is why fluorescence always has a higher wavelength than absorbance. So as you can see on this, they say the fluorophore, which is like a fluorescent dye, the room temperature are predominant in the lowest vibrational level of the ground state. Basically, it's this kind of solid black line. Those are kind of molecule at the ground state. And then therefore the energy for absorbance, they can jump in now from S0 all the way to S2, or S0 all the way to the first vibration level for the S1, or maybe the third, fourth vibration level for the S1. So they can go up anywhere, all right? So basically your energy transition here, the higher the gap, meaning that they have a high energy, all right? So your fluorophore will able to go to different kind of energy level here for absorbance, all right? As compared to the fluorescence, as we already know, even though your molecule is in S2 or your molecule in S1, those electron is going to undergo an electron internal conversion. All right, they are not going to emit the fluorescence from S2, or they are not going to emit the fluorescence from the highest energy level for S1. They prefer to release the fluorescence at the lowest vibration level for the S1, which is a solid red color 9 here. And that's why your fluorescent emission predominate from this kind of the lowest vibration level or the first excited state. And then they may reach any vibration level of your S0. So they can go all the way to the black dash 9. They can go the second one, they can go to the first one, they can go anywhere they want. So this kind of the green, or kind of like a little bit blue or green colorish fluorescent line is always shorter than the absorbance line. And that's the reason why, if your energy gap is smaller, definitely your wavelength is going to be bigger, all right? So energy and wavelength, they are anti-correlated, right? Bigger energy, you have longer wavelength. And that's the reason why the fluorescence always happening at a longer wavelength than the absorbance, because absorbance has usually can have a high energy transition. Fluorescence also has a short energy transition on that. All right, so this phenomena, we call this a Stokes shift, and this is the second phenomena for the fluorescence. And the third phenomena for the fluorescence is that they say the fluorescence spectrum or for emission spectrum usually has a mirror image to the absorbance spectrum. And again, we still go back to this energy transition here. So in the S0, same as before, is a ground state, and S1 is excited state, and those station 9 are different kind of vibration level in excited state. And then you can see on that if a molecule, let's say they're starting at the black color 9, which is ground state, they were able to transition to the excited state, and they can 0 to 0 prong, or 0 to 1 prong, or 0 to 2 prong, or 0 to 3 prong. They can vibrate, they can absorb the light into different kind of energy level on that. And that's why you can see in the spectrum on the left here in the corner, this is a solid 9 is absorbance spectrum. So if you have a high energy transition, that will represent 0 to 3 prong, which is a shorter wavelength, and 0 to 2 prong, which is the middle of the wavelength. 0 to 1 prong is a longer wavelength because their energy gap gets smaller. That's why the wavelength gets bigger on that. So your x-axis in this plot is a wavelength, all right? So this is typically the solid black color 9. This is absorbance spectrum. And it's because your molecule can transition to different kind of excitation state in a different kind of energy level. But again, for the green color one, as you can see, the energy diagram from the right, they were able to release the fluorescence. And these fluorescence I'm talking about, they usually happen at the first vibration level of the excited state, and they go to the ground state, like 0 to 3 prong, or 0 to the 2 prong, or 0 prong to 1, and then 0 prong to 0, all right? So basically they were able to, same way as we see absorbance, they were able to move down from the highest one to the lowest one. And once they're moving down, as you may imagine, on the larger energy gap, meaning that they are going to have a shorter wavelength, all right? So that's why the 0 prong to 0 basically is going to overlap with your absorbance. But your 0 prong to 1 is going to have a longer wavelength than absorbance, because this 0 to 1 prong energy gap is a little bit shorter than absorbance. And 0 to 2 prong, they're going to move longer and longer because this energy gap gets smaller and smaller. And that's the reason in the end, you will see on that there's a mirror image between absorbance and emission, fluorescence emission spectrum. And because this kind of different energy level for this particular molecule on that. All right, so this is the reason why the fluorescence usually has a mirror image or then absorbance spectrum on that. All right, so since we're talking about the fundamental knowledge about fluorescence, of course, nowadays, people has been widely used a lot of this fluorescence technique to detect a lot of maybe the biological interaction or maybe some chemical interactions. So here are just some example I want to demonstrate. So there are about like 3 different kind of technique has been widely used nowadays. And some of them even though, can be used for the pharmaceutical company. For example, nowadays we want to find out the cure for the COVID-19, right? And then the pharmaceutical company now they put a lot of small molecule, synthesize it on it from organic chemistry. And then how do they know this molecule can bind or can kill the virus? And one way they can do is they can use in the fluorescence anisotropy, because those small molecules they synthesize from the lab usually has a fluorescent characteristic. And once they have fluorescent characteristic, this molecule, when they bind into the virus, they may change a different kind of property there, and they were able to detect what is the binding affinity between this drug to this virus. And then they were able to see if this has a very, very tight binding, meaning that they were able to maybe kill the virus, all right? So anyway, so these are all the 3 different techniques, and we'll talk about one at a time. The first one is the fluorescence anisotropy, and we'll talk about that later on. And the second one, which is a very famous one, is called fluorescence resonance energy transfer. They usually refer to as a FRET, right? F-R-E-T, FRET. And this basically is energy transfer between the 2 different kinds of the fluorescent dye. And the third one is the fluorescent microscope, and I know you guys may already hear from Dr. price talking about the EM, but this is a fluorescent microscope, which can also be very, very powerful. They were able to visualize, for example, can visualize a cancer cell in the living cell, and they can visualize on it, and they can see how the molecule moving around inside the living cell. So these are all the 3 fundamental fluorescent technique has been widely used, and let's take a look one at a time. So the first one is a fluorescent anisotropy, all right? So anisotropy is a little bit difficult to understand, but you guys may know the isotropic. Isotropic meaning that it's equally distributed, all right? So your molecule is tumbling around very equally, and then we call this as isotropic. If a molecule, you know, one direction has stronger than the other one, we call this as anisotropy, all right? So here is one phenomena you can see on that on the top here, basically is a ligand, and this ligand has put some fluorophore, basically has a fluorescent dye on this ligand. And then if this molecule is, you know, rotation or translation or, you know, vibration very, very quickly inside the water solution, then, you know, when you shine the light on this molecule, we can call this has a very, very low anisotropy, or it's called isotropy, because those molecules are fast tumbling inside the solution. Then even though you have a polarized light into this molecule, then the molecule will, you know, dissipate this polarization there. Therefore, it's low anisotropy. On the other hand, if you have a bigger molecule, let's say it's a virus, all right? If the bigger molecule is a virus or some protein molecule, this big molecule, when they bind into this ligand, and then the whole molecules has been slowed down the rotation or slow down their vibration more, because molecule is very, very big, it's a very, very giant, and the rotation is slowed down. And when you shine the polarization light on this molecule, interestingly, this molecule can still preserve this polarization layer. Therefore, we call this is a high anisotropy. All right, so that's what I'm talking about. Nowadays, people want to figure out how can we cure the COVID-19, or how can we cure the coronavirus. And one way they can do is they can synthesize this small ligand, and this ligand usually carries some fluorescent signal. And then if this ligand were able to bind into the virus, then they were able to slow down the motion of this ligand, then therefore you were able to see this high anisotropy. And you can use this as a way to detect the binding affinity between your ligand and your virus, all right? And this has been widely used in the pharmaceutical company nowadays. So anyway, this fluorescent anisotropy, as you can see in the formula here on the bottom, there is a parallel intensity subtrate a perpendicular intensity. So basically, this is not usually you will see the fluorescent. Your fluorescent need to have some polarization there. You need to have some parallel polarization and perpendicular polarization. And this parallel or this perpendicular polarization, when they shine to the molecule, if the molecule can still preserve this polarization, then we can call they have a high polarization there, or they can have a high anisotropy there. And compared to molecules, the fast tumbling there, then you may not be able to see this polarization there. So that's why you can see the note I saying that usually those are fully solution, a fluorophore or maybe some fluorescent dye, they usually rotate about like 50 to 100 picosecond. So picosecond is 10 minus 12 seconds, all right? And the lifetime of this fluorescent dye usually is about nanosecond, 1 to 10 nanosecond, which is very, very, very short, right? But still, the picosecond is faster than the nanosecond, all right? So your molecule is way faster than the lifetime of the dye. Therefore, you are going to release this kind of the polarization there. That's why in the end, you have a low anisotropy for this molecule. As compared to in the bottom here, if there is a protein molecule or there is a virus molecule bound into this kind of ligand, then the rotation for this molecule is going to slow down. Originally, it's a picosecond range on the top, but now they become like a 15 nanosecond, all right? They're slowing down. And this nanosecond is pretty much the same as your lifetime of your fluorescent dye. Therefore, you were able to see the anisotropy signal because they were able to detect by the machine on that. All right, so this always just monitor, the molecular tumbling. The molecule is super big. They were able to slow down the rotation or slow down the vibration of the molecule, all right? So one of the example here, we can see on that, this is one of my previous research, all right? So in that time, we tried to identify the binding of the molecule to the protein, all right? So you can see this is SRP43. This is one of the protein molecule, and this molecule going to bind into a small ligand, and this ligand basically TM2L18, so this is a smaller protein, all right? And this protein, I put a fluorescent dye on it, And what I did is I titrate the different amount of the protein molecule, and I monitor the anisotropy change. As you can see, this red color 9, because if I increase in the concentration of protein, and protein is very, very big, all right? And then this small ligand is very, very small, like maybe 1 or 2 kilodalton. It's very, very small. And once you titrate the big molecule in, your anisotropy signal going to increase because you slow down the molecule motion, and that's why anisotropy signal going to increase. But of course, people may argue on that. How do you believe this signal increase is represent of the binding? It could be some artifact, like some impurity, just randomly, binding to your smaller ligand, right? So it could be that. This always happen in nowadays, right? In the peer review journal, if you submit that, all the reviewer always criticize all your data, all right? And of course, I already know that they are going to criticize on it, so you need to do some control experiment. So one way to further test this result is we can preform a complex, as you can see in the panel C here. Panel C basically just tell you that if I have a big protein, all right, this big protein is like a pink color protein, they bind into those green color ligand, which is the TM2L18, they preform this complex. And then once they preform the complex, the fluorescent dye is on the ligand, all right? So now in the beginning, you have very, very high anisotropy because the whole molecule is very, very big. It's going to slow down motion. But once you chase the complex with a smaller ligand, which is L18, and it's very, very tiny, all right? Only 18 amino acid is very, very small ligand. And this ligand, you keep adding a huge amount of this ligand, they were able to compete the binding to the protein, and then in the end, your protein going to kick away this kind of ligand, and then your L18 ligand is going to bind, as compared to your fluorescence label ligand is going to dissociate. And in that case, if this ligand get dissociate, as you may imagine, because those ligands were able to moving around very fastly inside the solution, and that's why anisotropy is going to decrease if they kick away by this kind of ligand. So basically, this is a competition experiment, all right? So that's why you can see the signal from the panel D here. In the beginning, I have a preformed complex. Preformed complex meaning that I have a ligand, fluorescent dye ligand, with my protein. They already bind. That's why in the beginning, you can see anisotropy is very, very big. But if I increase in the competitor concentration, which is L18, and this competitor basically is no fluorescent dye on it, so you will not see any fluorescent signal. The main fluorescent signal is coming from your TM2L18, this ligand. And then you chase away by this kind of unlabeled ligand, then the signal is going to decrease on that because they will kick away from binding to the protein. And then this fluorescence labeled ligand is going to increase the motion in the solution. And that's why it's become low anisotropy signal on that. And these two experiments can further justify fluorescent anisotropy we can use as a way to detect the binding affinity between your small ligand to the big protein molecule. All right, so and the other famous technique here basically is called a FRET, all right? So it's fluorescence resonance energy transfer. And this also has been widely used in a biophysical, biochemical study on that. Even some chemistry, they're using this study for some small molecule, they can also apply the FRET. So basically FRET is energy transfer. So let's say you have one big protein, all right? This protein has one fluorescent dye on it. And also you have the other ligand. This ligand also has a fluorescent dye on it. You have two fluorescent dye and one at each molecule. And today I want to study the interaction between these two molecules. You can imagine one could be the drug, the other is the virus, all right? And I want to study the virus interact with this drug, how the drug bind into the virus. You can put one dye in the virus, one dye in a drug, and then you are going to detect interaction between these two dye. So one way you can, once these two molecules going to bind together, form a complex, as you can see on the right, and then once they form a complex, the two dye are in a close distance. They are close plasticity on that. And once in a close plasticity on that, interestingly, if I excite the molecule on the other one of the dye, and this dye is not going to fluorescence because they are in a close distance, they are going to transfer energy to the other dye. And once the energy has been transferred to the other dye, you were able to detect fluorescence coming from a different dye. All right. So this has been widely used to detect the molecular distance. All right. We can call a molecular ruler by using a FRET assay. All right. So basically, the reason why the two molecules are in close distance, why they were able to undergo an energy transfer, and here is a diagram I'm going to show on that. So we have one dye called a donor dye, and the other dye we call acceptor dye. All right, and donor basically is the one going to excite by the light. All right, and once you excite by light, as you can see this donor at the blue color line, once you, put a blue line there, they absorb the fluorescence, absorb the light. And once absorb the light, usually, without, if let's say today, without acceptor dye, your donor dye is going to emit the fluorescence, which is the green color one, without any problem, right? If there's no energy transfer. On the other hand, if today, let's say you have the other acceptor dye in a close distance, but with the donor dye, all right? So when the acceptor dye is present, which is right in the right, and interesting on that, instead of release the fluorescence from the donor dye, they are undergoing this kind of energy transfer. So they will transfer the energy into the acceptor excitation state. And once in the acceptor excitation state, this energy is going to release the fluorescence from acceptor dye. So then you were able to see the fluorescence show up in acceptor wavelength instead of the donor wavelength. And this can consider as a fluorescence resonance energy transfer. And as I say now, this is highly distance dependence. If the two dye are in a close plasticity, then you will able to see this signal. As compared to two dye are far away, then you will not able to see this signal, all right? So I'm talking about like 50 Armstrong distance, all right? It's very, very short, right? All right, so the other phenomena you can see on that here is if I have two dye, again, I'm talking about a fray. If the two dye, and then if these two dye are in a close distance, as you may imagine, they are undergoing a fray transfer. So the donor dye, the solid 9 basically is emission spectrum, the dash 9 is excitation spectrum, basically they form the mirror image as we were talking about before. And let's say donor dye, acceptor dye, if they are in a close distance, and also their, spectrum are highly overlapped. So you can see the donor emission spectrum is highly overlapped with the acceptor excitation spectrum. All right, and this meaning that they were able to using the donor, emission energy directly transfer into the acceptor excitation energy, and therefore your acceptor were able to emit the fluorescence. And that's the reason why every time when you do this kind of experiment, you need to make sure the selection of your dye need to make sure they have a spectrum overlap between emission wavelength of donor and acceptor for the excitation wavelength. All right. So here's one more exaggerated example here. So the one on the left is the one without any thread. So basically, you have a box, and this box, as you can see on that, there is a blue color dye, and it also has a red color dye. All right, and if these boxes are are far apart each other, it's very, very far apart. They are not close nearby. Then when you shine the light on your blue dye, so basically you're going to see the blue fluorescence show up. And also you will see a little bit of fluorescence show up in the red dot, but it's very, very low compared to the blue color one because they are far away, all right? But interesting that if you can see the right, if these boxes are close, all right, they are very, very nearby. Then when you shine the light on the blue dye, and interesting on that, instead of emit the fluorescence from the blue dye, they are going to undergo energy transfer to the red dye, and therefore your red dye is going to show the fluorescence. That's why you will see the red fluorescence increase. So typically, you will see on that is if there's a fresh signal show up, your acceptor dye fluorescence will increase as compared to donor dye, your fluorescence will decrease. And that's pretty much the signature of the fresh signal.
Liang?
Yes.
Is it correct to assume that the area under this graph would be like the total energy like going on inside this transition, or is that not correct?
I think you can say that. You can say that. But you need to make sure there's no other, energy or release. All right. You need to make sure there's just pure energy transfer. There's no other side effect from these kind of reactions. Definitely, the area will be the same for both.
Okay.
Right. So people use this as a way to monitor the distance of the two molecules because sometimes the molecule in a solution. How are you going to measure the distance? And one way they can do is put a two dye one each molecule, and then if you see the signal show up, they will able to serve as a way to calculate the distance between the two molecules. So that's why I'm saying that they call this is a molecular ruler, all right? And then of spectroscopy, a ruler. So if the donor is acceptor, if they are in a close distance, and then you can see on the acceptor signal, which is the red color one, it has a very, very high signal. As compared to, the donor acceptor, if they are far away, then you can see the red color peak has been decreased, as compared to the green color one has been increased. And basically, this is happening between the donor and acceptor dye of the energy transfer. And then people usually report as the R0. So R0 basically is a number like roughly about 50% of energy transfer, and then represent about their the distance between the two die. And usually the transfer efficiency usually about like 3 nanometer to 7 nanometer distance. All right, so it's very, very small. And people usually use this as a way to monitor the distance between the two protein molecule. All right, so it's been widely used in a lot of study by using a thread. All right, so if you're interested, you can always check on Google on that. All right, so far we've been talking about the fluorescence. All right, and then of course you may wondering how are we going to put a fluorescent dye into the molecule? All right, so some molecule, you know, intrinsically they have fluorescent property on that, but some protein molecule, for example, they don't have fluorescent signal. How are you going to put the fluorescence into the protein molecule? All right, so here's one example. They can put a fluorescent dye in vivo, or in vivo meaning that it's inside the organism. All right, so we can even study the cancer cell inside maybe the rat or inside the mice. So how are we going to label these cancer cell inside the mice? So one way they can do is using some genetic technique. All right, so sorry about that. This may be very biology. All right, so for those non-biology major, you may not able to understand this very well. So basically, they call this is amber suppression. All right. So what they're going to do is they can put some non-unnatural amino acid, and this unnatural amino acid basically has some fluorescent characteristic on it. And this unnatural amino acid, basically, what they can do is they were able to carry by some tRNA, and this tRNA basically what usually is one can carry some normal natural amino acid into your ribosome, and ribosome were able to synthesize the protein right inside the cell. But today, if they introduce this orthogonal amino tRNA synthetase enzyme and also this orthogonal tRNA, and they were able to carry this amino acid or carry this fluorescent dye into your protein. And then basically these orthogonal tRNA synthetase, they were able to recognize your amber codon. Sometimes they refer to as amber suppression. So amber usually people refer to as a stop codon, all right? So when your protein synthesized in your body, when they see the stop codon, usually the ribosome will dissociate and then they will terminate your translation. But in this case, your tRNA synthetase were able to recognize the stop codon, and then they were able to deliver this fluorescent dye into the protein, and therefore you were able to label the protein with a fluorescent dye. All right, so this is very specific topic. All right, so I'm not going to talk about very detail, but if you're interested, you know, we can have some discussion later on that. And typically, in the chemistry way, how the people, you know, put the fluorescent dye into the protein molecule or into some organic molecule. So basically, they can use in the metal imide chemistry. So this is a metal imide fluorescent dye. You can usually purchase commercial right now. And then this metal imide functional group, basically, they will able to serve as an electrophile. And then your thiol can serve the nucleophile. And this thiol you can also find in a protein molecule, like cysteine molecule. And this thiol, they were able to nucleophily attack, you know, this double bond here. And then therefore they were able to form a covalent bond between your fluorescent dye and then you conjugate a molecule. And that's why you were able to label the protein. This will be in the test tube, all right? In the test tube with a fluorescent dye. All right, so this is one of my previous data as well. I want to study the interaction between two proteins, all right? And then how am I going to study on that? I want to use in a thread to study on that. So I label one dye with I think Alasa 488, this is donor dye. And the other dye labeled with the other protein, I call it TMR dye, all right? So basically, if I have a donor only, which is the blue color one, you can see there is a fluorescent signal show up. This is a fluorescent emission spectrum. And if I have acceptor only, which is the black color one, all right? So basically they are highly separate. They are not overlapping each other because this is just emission spectrum. But of course, your donor emission is going to overlap with acceptor excitation wavelength, but their emission for both of them are separate. But if I have a mix of these two proteins together, and interesting, you can see on that donor signal has been decreased as compared to acceptor signal has been increased, all right? And of course, this probably is not very proportional increase on that, all right? You may see this increase is very, very small, and this decrease is very, very huge, all right? Because the energy transfer may not be very, very efficient on that. And then this is kind of like example to show out if two dye are in close distance, and then they were able to undergo this energy transfer, and that's why you were able to see this kind of thread. All right, so the last one is the fluorescent microscope, all right? So fluorescent microscope has been widely used. Of course, you can see on that they can visualize some living cell. they can use in this, put a sample, a specimen, label with some like antibody fluorescent dye, and then they were able to specifically look at the cell, and they can even see the nucleus on that, all right? So people have been widely used nowadays, all right? They can even do a single molecule in a living cell. So I'm not going to talk about very detail, all right? So, but you know, people can also use in this fluorescent microscope to look at the three-dimensional structure of the phenomena or some chemical reaction inside the cell. So this is called super resolution microscope, and this is one of Nobel Prize nowadays, all right? So the stone is one of the techniques they are using right now. They were able to capture the super resolution for the particular protein molecule inside a living cell. All right, so we still have only 5 minutes left, and then I'm going to finish the part 2, all right? So part 2, basically, usually I will not talk about this because usually just part 1 is enough, but part 2, since we have online here, and then I thought maybe we can talk about some of the principle of protein purification. So for those of you who already took my biochem, you already know this. For those of you who haven't taken a biochem, or is going to take a biochem, you may see this again, all right? So the reason why I talk about this because, as you know, what is the most famous topic nowadays? COVID-19, everywhere, every day, all right? So scientists now has been spent a lot of money, a lot of time want to develop a vaccine or want to develop a drug to kill this virus. But how they are going to do it, as we already know, and anisotropy we are talking about, they can use that one to detect the binding between virus and some ligand. But of course, before we study this virus, there are some of the signature feature on this virus. This virus has other protein there, all right? So you can see there's E protein on the top of the surface, and this E protein stands for the envelope protein. There's S protein basically the spike protein, and there's M protein referred to as a membrane protein. So there are a lot of protein molecule on top of this virus surface, all right? And then, of course, how are we going to study this protein? And one way we can study is we can purify this protein in a test tube, and then we can study the interaction with some particular drug or some particular molecule. All right, all you can do is just isolate the virus from maybe the patient in the hospital, all right? But you need to be well prepared on that, all right? So basically, this is kind of protein molecule is a building block of all organisms, all right? Even our human body, we have thousands, billions of protein inside our body, can help us, you know, maintain our daily life. And those are because those protein molecule, those enzyme molecule, they will perform specific chemical reaction inside the body. All right, so talking about the protein molecule, there are different levels of protein molecule. You know, protein structure is very, very complicated. You don't need to understand this. I don't expect everyone memorize this, all right? Just that you know on that, protein molecule is very, very difficult molecule. It's very, very big molecule. It's very hard to understand it, how they interact with different molecules. And of course, in order to study this protein molecule, usually we will do in the lab is we can use in some bacteria or using some yeast or using some different kind of organism to express the protein. And then once the protein is expressed, as you know, inside the bacteria or inside the yeast, there are tons of the protein there, and then you need to fish out your target protein. And one way to fish out your protein is you need to go to the protein purification, all right? And this purification can make sure in the end your protein is very pure. Then you can study this protein-protein interaction by using the fluorescence technique we have been talking about. So these protein purification, usually they can undergo in cell disruption, lysis the cell, and then once the cell lyses, you can use centrifugation, you can remove those unbroken cells. And then once the cell is broken, or you can do some column chromatography, and in the end, you can do some quantity and quality analysis on that. But today, I'm not going to talk about everything. I'm going to talk about some column chromatography, all right? This usually is the most interesting one, how to purify the protein. So we know protein molecule has different kind of amino acids. It's a polymer of amino acid. And those amino acids have different charge, different size, and different solubility, different hydrophobicity, different thermal stability on that. And one way we are going to purify, we need to use in this kind of physical and chemical property. And then that's the only way we can purify this protein out from different kind of impurity. And of course, people usually do some column chromatography, as you already hear from maybe the OChem, organic chemistry, already talking about this. So this is affinity column. As you can see, there's a reservoir here. And this reservoir, they can put some of the molecule inside. You can put a protein inside. And then your protein sample, once it go to this column, as you can see, there's a mobile phase, so they can moving around. That's called a mobile phase. And inside this column, there's a stationary phase. And this stationary phase usually has some like a solid matrix inside. And this matrix will help you to bind into the protein. And that's why when you load the protein inside, and there are different protein inside this ABC protein, but in the end, as you can see on that, molecule A has a little bit tighter binding than the molecule C, because molecule C almost coming out, but molecule A is still inside the column because they can retain the protein a little bit longer, and they can bind into the protein a little bit longer, and therefore you were able to separate between A, B, C molecule. And that's why in the end, I can just collect the molecule A and then can get a very, very pure molecule in the end. And you may be wondering how I'm going to get A, if A is binding there forever, how I'm going to get rid of that. So usually people can use in some high salt, because usually it's electrostatic interaction, and you put a high sodium chloride concentration, then your molecule A may be out of the column easily, all right? And of course, you need to make sure your impurity has been washed away before you elute your target molecules. And the other one, people usually use a lot is ion exchange chromatography with cation exchange, with anion exchange, and even myself, I use a lot of cation exchange. And cation exchange basically is your resin or your solid support is a negative charge. Your protein you're interested in or your molecule interested in is a positive charge, then they can bind by electrostatic interaction, and then you can wash away impurity, and therefore you can use in the high salt, then elute the protein out of your column. And the other one is the size exclusion chromatography, basically using the size, all right? So different molecule, different size, we have some kind of polymer, and this polymer inside this column basically has some cavity inside. And this cavity will trap small molecule. So big molecule, when you load into the column, they will come out immediately, because those big molecule like green color or like orange color ball, they will not go to this small cavity. As compared to those purple color ball, which is a small molecule, they are going to trap inside the column, and therefore they are going to have a longer retention time than those big molecule. And that's why it will separate molecule by different size. And this we call a size exclusion chromatography. And the other one is affinity chromatography, as we're talking about. The protein molecule can bind, and once they bind, you can put some like a ligand or some competitor, then your protein molecule will come off on the column, all right? So I'm not going to talk about this very detailed. So basically, this is my last slide, all right? So we have talking about anion exchange chromatography, cation exchange chromatography, and size exclusion chromatography. And basically, anion exchange chromatography, your stationary phase, which is your polymer, is going to be positive charge. And that's why your protein of interest in or your molecule of interest in, they were able to bind into this positive charge. So your protein interesting is negative charge, and they can bind together, and those impurity, they can wash away, and in the end, you can use in the high salt to elute out your protein. And of course, cation exchange will be different than anion exchange. They can use in the stationary phase, it's a negative charge, but your protein interesting or your molecule interesting is a positive charge, and they can bind each other. And then your molecule in the end maybe elute out of there very quickly, which is a negative charge, and they can use in electrostatic interactions. And the last one is size exclusion chromatography. As I already saw in the picture before, they can separate the molecule in different size. Small molecule can trap in the cavity, big molecule can come out of the cavity, and that's why you can separate by the different size. All right. So again, for those of you who are going to take a biochem, I'm going to text you guys this. But for those of you who want to take biochem, don't worry about this. All right. So this our technique has been widely used nowadays to purify different kind of molecule or purify the protein molecules. All right. So again, this is my slide, all right? So I appreciate everyone to attend this workshop, all right? So hopefully, everyone enjoy this workshop, and then I wish everyone has good summer research here, all right? So if you have any question, welcome to send me an email or welcome to ask, all right? So you guys enjoy your summertime.
Thank you, Dr.
Thank
Liang.
you.
Thank you.
Thank you.